Okay, so we are going to talk about now in detail the Polycomb group and trithorax group in the context of epigenetic cell memory. What we just learned is that cell fates, they are established early during development, and they are different cells. They become specialized due to by thorax complex and antennapedia complex in fruit flies, and their expression pattern, for example, ABDP, ABDA, UBX, they get established in specific segments of the body, and they are maintained all the way till that. So this is the adult, this is like 2 hours old embryo. And once the expression pattern of UBX, which gives identity to halter, or absence of UBX gives identity to wing, they are maintained. For example, this yellow one are the wings where UBX will be off, and here this is the halter, this is the tiny appendage in the adults, which is necessary, which is important for maneuvering the flight of the fruit flies. Now UBX is on in the halter, and this expression pattern, which is established early on in the region and eventually in the imaginal disc in the halter, where UBX is on, it remains throughout on, and this expression pattern is maintained in the on state by trithorax group in halter, but it is maintained in the off state by Polycomb group. So the Polycomb group, or the because as the word indicates, it's a group of proteins. And it contains multi-protein, megadalton, big complexes of proteins. And this group was founded after the protein which is called Polycomb. Polycomb was discovered based on the phenotype, not the gene, but the Polycomb phenotype was discovered by Ed Lewis, who won Nobel, I think in 1998 or so, together with his wife, who was a technician in his lab, Pamela Lewis. So they both discovered this phenotype, and the phenotype is basically in Drosophila males. So I'm drawing the 3 pairs of legs. There is one specific appendage, the comb-like structure, the polycomb. So these combs, they are called sex combs. They are present only on the first pair of legs, not on the second and third, only in the males. And they use these structures to hold the female during mating. Now, in the Polycomb mutants, what Ed Lewis found that the second and third pair of legs, they also, in the Polycomb mutant, although the first pair had sex combs, but second and third pairs also got extra, and this phenotype is called extra sex combs because they are normally absent in the wild type. In rest of the forward genetic screens, then the genes in those mutant screens which led to such Polycomb phenotype, they were grouped as Polycomb groups. Okay, Polycomb group proteins. Later on, what we have learned that the presence of sex combs on the first leg in males in wild type is due to one of the gene in antennapedia, which is called sex combs reduced. Okay. So this sex combs reduced is only expressed in the first leg, but it's off, it's silent in second and third leg. You remember the differential gene expression? Although cells in second and third leg, they contain same genome, same chromosomes. All the gene set is same, but due to differential gene expression, this gene is off in second and third. And it's remained off due to it's kept off by Polycomb group. This is what we learned in last 30, 40 years. So once you have mutation in Polycomb group genes or Polycomb, okay, we saw activation of sex chromes in the second and third legs, and we refer to it as extra sex chrome phenotype. Now it became clear that Polycomb is involved in maintaining homeotic genes in the off state when cell fates are determined, and these cell fates are to be perpetuated or passed on or inherited during process of epigenetic cell memory. Polycomb is involved in maintenance of silencing of sex chrome produced or other homeotic genes, like I said, UBX in the wing disc, in the wing cells, for example. So once they learned about Polycomb mutants, another set of forward genetic screens, they were interested in finding suppressors of Polycomb, which means genes, when they are mutant, they suppress this extra sex chrome phenotype. And what they discovered, the suppressors of Polycomb, they were called trithorax group. Because if you have Polycomb mutation over trithorax group, the extra sex chrome phenotype is suppressed and you have wild type. Because because the ectopic activation of homeotic genes in second and third leg due to mutation of Polycomb. So Polycomb was supposed to maintain sex chrome produced silent here. Now it gets ectopically activated away from its natural position of expression. This ectopic activation is due to action of trithorax group. We said they are activators. They maintain genes in the on state. So absence of Polycomb led to activation of homeotic genes ectopically due to action of trithorax. Now if you have trithorax also mutant, so minus trithorax over minus PC, the ectopic activation is gone and only wild type sex chromes are there. So that was the discovery of Polycomb and trithorax groups of genes. Initially they were just, because we did not know the molecular function of these genes, just based on these forward genetic screens, the genes which impacted extra sex chrome phenotype by having extra sex chromes, they were grouped as Polycomb. The ones which suppressed extra sex chrome phenotype, they were grouped as trithorax group genes. Now, what is actually function of Polycomb and trithorax? It was only, in 1992, I believe, it was David Hogness' lab, together with Renato Paro. I know this for sure because Renato is my mentor. Okay? I have read this paper. So this phenotype or mutant was discovered in 1963 or 64 by Adler's lab, but the gene for Polycomb was cloned in 1992. And once the gene was cloned, it was only then we learned what kind of domains or structure this protein may have. It was not a big protein. Polycomb was like 300 plus amino acids. But what they found that it shows homology to another protein which is called HP1. So this is Polycomb. Okay. So the protein sequence of Polycomb, it had a specific domain amino acids which is called chromodomain, which was present in heterochromatin. So a protein heterochromatin protein 1, HP1 of fly, which contains also towards C terminus chromo shadow domain. This is very important concept. HP1 is involved in maintaining the constitutive heterochromatin region, which is always containing, chromosomes in highly compact or condensed form, gene poor, transcriptionally inert, and HP1 is known to play a crucial role in maintenance of constitutive heterochromatin. Now, since it was already known that this is involved in maintenance of heterochromatin, in presence of chromodomain here, similar to HP1, it led Renato and David to propose that Polycomb is a chromatin binding protein which is involved in silencing, which was already known from genetic evidence, but this was first evidence emerging that it is chromatin binding protein, most likely. But chromoshadow domain was missing in Polycomb. Chromoshadow domain means these are similar amino acids in HP1 but with slight differences. Now, in Polycomb group, other proteins which were known are, for example, there was a protein called enhancer of zest, posterior sex chromes. All these were names of the genes which influence the extra sex chrome phenotype. Polyhomeotic, and so on. Okay? Trithorax group it comprised of, for example, trithorax, ASH1, Gaga factor, CBP, there are different proteins, Brahma. So these two groups of genes, there were like 18 to 20 different genes in trithorax initially. I'm talking about 30 years ago, and 18 to 20 genes, for example, in Polycomb group. But the molecular function was not known. It was not known what they are doing at the molecular level. Just the empirical evidence was that, the Polycomb group, they lead to, anterior transformation. When I say anterior transformation, the legs, they become, for example, the second and third one, they become similar to the first pair of legs. Okay? And similarly, if you have mutation in Polycomb, the ABDB, which is restricted only here because ABDB is maintained in silent state in abdominal segments, in the wing, etc. ABDB spreads. Okay? So expression of ABDB in Polycomb mutants will become more towards anterior. So it was known that Polycomb group is involved in maintaining homeotic genes in the off state, and that's how they are involved in maintaining the epigenetic cell memory. Okay, so the cell fate, which is established early during development by establishing gene expression patterns of homeotic genes in a very characteristic manner, they are maintained in the off state by Polycomb and on state by trithorax. However, it was not known that what is the detailed or what is the mechanistic pathway through which Polycomb and trithorax they exert their gene function, they exert their molecular function. However, what was known when they started, back in late 80s and early 90s, when DNA sequencing became a bit handy, a bit easier, it was still very difficult to run those sequencing gels, but nonetheless, they could, sequence after cloning these genes. They could sequence, and all they could do was, they could do homology and then see what kinds of domains are there, like we just saw in case of Polycomb. They saw this chromodomain, which is similar to HP1. HP1 is a heterochromatin protein. It's a chromatin binding protein. So Polycomb maybe is involved in binding to chromatin physically and maintaining silencing. So based on these searches, what they learned that Polycomb and trithorax proteins, they contain, another specific domain which was called for example, just like HP1, remember, we talked in great detail about HP1 and discovery of HP1, that it was discovered in suppressor of variegation screen, a forward genetic screen carried out in Drosophila for position effect variegation. Another protein which was discovered in forward genetic screen for position effect variegation was called suppressor of zest. And the short form is SUSI. Not suppressor of zest, not suppressor of zest, suppressor of variegation, because it was position effect variegation screen. And this protein, it contains specific amino acids which was conserved in, for example, some and not all of Polycomb and trithorax group proteins. And this domain of amino acids was called a set domain. Set domain because these amino acids, this specific domain of amino acids, it was present in SUAR 3-9, which was discovered in forward genetic screen for position effect variegation, which was involved in understanding the constitutive heterochromatin. It was also present in enhancer of zest, which is short form is EZ. So for example here, enhancer of zest, EZ. It was present in even a trithorax group protein, which is called trithorax. So that's why the domain is called set, SUAR. So S for SUAR 3-9, enhancer of zest, E, and trithorax. Set domain. Later on, we have learned it's also present in ASH1, which is another trithorax group protein. And for long, the function of set domain was not known, although it was known that trithorax and Polycomb, they associate with chromatin. They maintain gene silencing or gene activation by associating with chromatin. And it was also known that all the genes which are bound by Polycomb or trithorax, all the targets of Polycomb and trithorax, they contain specific sequences which were named Polycomb response elements. So, for example, if this is a gene, UBX, let's say it's UBX gene, it was discovered that Polycomb and trithorax, they were initially named Polycomb response elements because they were initially discovered as Polycomb binding sites. So if this is the UBX promoter and the gene, for example, this is the UBX gene, these Polycomb response elements, which are named as PREs, Polycomb response elements, they are present in the vicinity of Polycomb targets. And it was clear if we interfere with these binding sites, these elements, cis-acting elements, the silencing of UBX due to Polycomb was impacted. So they gave the initial clue that Polycomb, and later on we also discovered that trithorax also shares this binding site. So that's why they are called PREs, trithorax response elements. It was clear that Polycomb and trithorax bind to these cis-acting elements here, and then they exert their positive, so activating, or negative, the silencing effect on the gene next to these PREs or TREs. So let's now quickly look at UBX, for example. We will now just focus on UBX gene. This is the haltered, the wing disc. This is the halter. UBX, for example, is off when the wing is formed, and UBX is on in the halter. We will consider this as a cell fate or cell memory marker here. Now, in case of Polycomb and trithorax, they, as I said, they are known to be bound by PRE elements. And in the homeotic genes, by thorax complex, in the antennapedia complex, they did find these cis-acting elements, which I refer to as Polycomb response elements. They were next to ABDB, they are next to ABDA, UBX, etc. However, the big twist came in, I think it was 2002 or so. The Thomas Genoan's lab in Vienna, they discovered that SUVAR 3-9 in Drosophila is actually a histone methyl transferase. And what they could pinpoint was that the methylation activity is due to set domain. And what they also showed specifically that histone H3 amino acid lysine 9 gets methylated by set domain containing SUVAR 3-9 in Drosophila. And what they could show that all the constitutive heterochromatin, the chromocenters in fruit fly, that is labeled trimethylated, lysine 9 trimethylated by set domain containing SUVAR 3-9. They also showed that in mammals, there are two homologs of SUVAR 3-9, and the heterochromatin is disturbed or collapsed in the absence of SUVAR 3-9. This discovery, it led to further characterization of Polycomb and trithorax, although Polycomb and trithorax were known to maintain epigenetic cell memory, because you remove Polycomb protein, you lose the cell fate, wing becomes halter. Wing where UBX is off, and halter where UBX is on. So if you have Polycomb minus minus, UBX will become on, and this on in the wing disc will convert wing into halter. And you will see bithorax phenotype. Or if you have trithorax here, minus minus, UBX will become off, just like wing disc, and you will see halter converting into wing. Okay, so these flies will have two pairs of wings. This one, the normal wing, normal wild type wing, and due to loss of trithorax function where UBX will become inactive as well and halter will become wing as well. So this was already known, but the molecular function or the mechanistic function of Polycomb and trithorax became really easy when the function attributed to the set domain was discovered in 2002, I believe, by Thomas Genowan's lab. It was a Nature paper. Go back and read Ree et al. I think it was 2002 Nature. There you will see they mutated lysine 9. You mutate lysine 9 to alanine, for example, which means alanine cannot be methylated and no methylation will be catalyzed by set because there is no lysine there. So that's how they did very beautiful analysis in which they showed that this is lysine 9 specific histone lysine 9 trimethyltransferase. However, conservation of set domain, as we said, set domain is present in enhancer of zest, which is a Polycomb group protein. Set domain is also present in, the trithorax group protein. People immediately try to understand what is function of EZ or trithorax. However, I would also like to mention that they also showed that this methyl mart is recognized by HP1. The chromodomain containing HP1, and this binding or recognition of HP1 is due to the chromodomain. They showed that, and HP1 was also discovered in forward genetic screen for Suvars in the suppressor variegation or position effect variegation screen. So this chromodomain, it helps HP1 to recognize trimethylated lysine 9 and constitute the constitutive heterochromatin. Here, after this discovery, people started characterizing EZ and trithorax about their function in set domain, what their set domains are doing. Because there also we have chromodomain containing Polycomb. So in the next part, we are going to talk about Polycomb and trithorax set domains with their molecular functions. Stay tuned.